Okay, to the stages of the meiotic division. To the stages of the meiotic division, we have the first phase which we call the interphase stage. So the interphase stage is a stage whereby the cells gets ready. We have what we know as DNA replication. So interphase stage is characterized by DNA replication. You have the replication of your DNA. In the cell cycle, we'll talk about cell cycle in our upcoming videos. In the cell cycle, the interphase stage is characterized by stages in which we have our gap1 stage. We have the G1 phase, which is the gap1 phase, and we also have the S phase, which is the synthesis phase, and we also have the G2 phase, which is the organelle formation phase. So all these phases are characterized in the interphase stage. But what we have in the interphase stage of a meiotic division is similar to that of the mitotic division. And like we've said, we have a DNA replication. We have the replication of the DNA in preparation for what we know as the prophase stage. So after the DNA replication, we have also organelles formation. We have organelles formation and growth. So we have the growth of the organelles that we up in the prophase stage in meiotic division or mitotic division. So we have the organelle formation, we have the DNA replication, which occurs at our synthesis phase, and the organelle formation occurs at your gap2 phase, which is the G2 phase. So after all this process, after the DNA replication, the organelle formation, and the formation of centrioles, the cell gets set to go into the next stage of division, which we call the prophase. And from the word pro means before. So it has this prophase, whereby we have the prophase 1 division. So the prophase here is called prophase 1 because, as I've said, meiotic division occurs in 2 stages, the first stage and the second stage. And you can see what's on the board now. The prophase 1 stage. Prophase 1 stage, what's happening at the prophase 1 stage? What happens at the prophase 1 stage is similar to that of our mitotic division, but I'll let you to know that there are some stages, there are some terms in which are hidden in this prophase 1 stage. In some texts, it is divided into early prophase or the middle prophase and the late prophase. But I want you to take note that the prophase 1 stage is characterized by what we know as by the stages I'm putting on the board here, the leptothene. We have the leptothene, and the other one, we have the zygothene, or in some cases they call it zygonema, and we have the pachythene. We have what we call the diplothene, and we have what we call the dihydrokinesis. And you know what kinesis mean from what I said. Kinesis, the root word is from kinetic, so it talks about movement. So the leptothene and the zygothene characterize what we call the early prophase. You can call this one the early prophase. You can call this the early prophase. And the pachythene or pachythene, you can call it the middle prophase. And the diplothene, with your diakinesis, we can liken it to what we know as the late prophase. So these are the stages that are involved in the prophase 1 division. So going on, we have the leptotin, zygotin, pakitin, diplotin, and the kinesis. There are some crucial things that happens in the leptotin stage and the zygotin stage, talking about the early prophase. What happens in the leptotin phase of prophase 1 is that you have the chromosome in a spindle fiber form. They are still in a fiber. You have the chromatin threads. You have chromatin threads formation in which chromosome is looking like a chromatin thread. Under the picture given through electron microscopy, you can see the chromosome are in a thread-like form. Replication has occurred, and you can see the chromosomes in a thread-like manner in our leptotin phase or the leptonema phase. You can see the chromosome in a thread-like structure, and with the diagram that I have on the board here, don't forget, we're talking about our leptotin. You have your nucleus, you have your cell membrane, and you have your nucleus, which contain the nuclear material. So you have your chromosome still looking like thread. Yes, you have your thread-like structure, and you still have your nucleus inside. So this is the leptotin phase, and you see that the chromosome are still in form of a thread-like structure. You have your cell membrane, you have your nuclear membrane, you have your chromosomes, which are thread-like, and you have your nucleolus. So that's our leptotin phase or the leptonema, and the next phase, which is the zygonema. The zygonema stage. The zygonema stage. What happens in the zygonema stage is that we have what we know as synapses. There is something. The synapses here is not the synapse that we have in nervous coordination. When we have the movements of impulses from one neuron to another neuron, there is a synapse between the two neurons. But the synapses here, which is on the board, is what we know as pairing up. Pairing. So synapses is characterized in the zygonema stage, and we have pairing of the chromosomes, and they become more thicker. So they become more thicker and visible when they are checked under, or when they are observed under the microscope. So this is the zygonema stage. I've talked about the leptonema stage. I know to avoid mise en place, which means to put everything in order. So I want you, and I believe you that you've drawn what's on the board, so that you avoid clumsiness of what we are doing. So these the diagram depicting a zygonema stage. So the chromosomes So the chromosomes pair up to form a thicker structure. You can see these are the chromosomes, and we call them homologous chromosomes. We call them homologous chromosome. You can see homologous chromosome. So when we move to the next stage, you'll understand what we mean by the picatinine or the picatinine, and you understand what we mean by the diplotin stage in the next video.